Good morning, everyone. I'm Laura Rudberg, MassBio's Director of Events. Thank you for joining us for MassBio's virtual Possible Talks. These are 15 minutes of inspiring and engaging content in the style of a TED Talk. With me this morning is Kevin Hrusovsky. He's the President and CEO of Quanterix and founder of the nonprofit Powering Precision Health. COVID-19 and the current pursuit to return to business as usual has exposed areas of great need and opportunity across the healthcare system. While many organizations have stepped up in the fight against the virus, the reliability of tests, treatments, and vaccines needed to truly reopen our economy continue to be called into question. PPH explores the intersection of these three elements against the backdrop of COVID-19. Kevin will detail how the PPH syndicate is harnessing cutting-edge biomarker technology to fuel critical advancements in early detection, enable deadly cytokine storm prediction, and deliver empirical insights into immunity that could help overcome testing challenges and set the stage for reliable, widespread home care testing. We have a few reserved minutes at the end for Q&A. I'm sorry if you see my children, they're trying to dance behind me and not be seen. We have reserved a few minutes at the end for Q&A. You'll see there's a little box at the bottom of the screen. You can ask questions throughout, don't wait till the end. And with that, I'm gonna turn it over to Kevin.
Thank you very much, Laura. This is a real pleasure to be with the Massachusetts Biotech Council. I'm a huge fan of Bob Coughlin and the whole team, and the impact that they've made in, I think, the Boston and the Massachusetts area has been profound. So we Quanterix, as well as our Powering Precision Health, really are excited to join today. The agenda I'm going to walk through is the pandemic is absolutely jolting a new reality throughout the globe. And I would maintain garbage in, garbage out as it relates to testing, and the public policy as a result of bad testing, I think, is under threat. The power of a global ecosystem to power precision health is being harnessed right now through a lot of our activities, and we'd like to talk about that and then describe the potential for uber-sensitive digital biomarkers today to help with COVID and tomorrow with overall transformation of home healthcare. So COVID clearly is having a devastating impact. Nearly 500,000 people worldwide have died, and we can see the number of cases is approaching 10 million. That is continuing to increase even at a somewhat elevated pace over the last week and a half. This slide just shows that the United States yesterday had an absolute record number of new cases. Some of that could be due to testing, but the record level of hospitalizations is also very concerning and somewhat independent of the testing predicate. But on the left-hand side, you can see the pace in the dark red states have actually increased their weekly number of cases by greater than 50% is the dark red. And glad Massachusetts is not on that, but you can see Florida and Texas are there, and California is quickly approaching. The good news is Massachusetts, I think, gets the gold medal, and I don't know if it's because the Massachusetts Biotech Council is here, but I suspect it plays a role. You can see that there's fairly significant declines in all the key metrics for Massachusetts, but when you look at the United States, you can see this second tier of ramp. And the only other country we can find around the world that's having that relapse rate is Iran. So this is a pretty delicate situation that we're currently in, obviously, and testing, I think, will be key to getting us out of this. The key part of this is that the devastation is not just due to deaths and all of the peril that it's bringing to families around the nation and around the world, but it's also the economy is starting to be significantly impacted. Trillions of dollars of pandemic costs are projected to impair the world, and we are seeing jobless rates that are unprecedented at this time. In the United States, you can see we're now projecting a fairly significant negative trend for our GDP. There's now estimates of -8%, even though this is shown -6%. Interestingly, the only country that we can really see around the world that seems to be not in a negative situation is China, whose number of people that have it is a very, very small fraction of the number of people in the country. The United States is now up to about 300 people per million. So it's clearly about 100 times greater than China. And you can see the unemployment rates are off the charts. It's approaching 20% in the United States. These are really levels of unemployment we haven't seen in the last 100 years, except for wartime, and we're actually approaching the massive depression levels. And you can see in the UK just how significantly the drop-off has been year to date in the number of hours worked. And you can see all these major announcements. They're still coming. And I know of some large corporations that are still talking about significant announcements over the next month or two of layoffs and furloughs planned, which is very devastating. Luckily, at Quanterix at this point, we've not had to have any because we became very central, and we've actually pivoted in helping the world around this crisis, which we're going to now talk a little bit about. Even though this talk is more about just PPH and the broader spectrum of what we call digital biomarkers, we will bring some specifics into it. So I actually think this is the intersection of politics and science that we've never seen in our lifetime. And it's a very big challenge for public policy when the science is still trying to catch up with really good testing. You can see some of these headlines are profound. We've had this Powering Precision Health Foundation that we founded 5 years ago. That's got several thousand of the top innovators in the world in it, as well as key policymakers. We also have a lot of investors from Wall Street, some of the largest in the world, attend these sessions, and many patient advocacy groups. So you really create a complete ecosystem that allows us to move public policy and innovations much more rapidly, trying to get these advances of the ultra sensitive digital biomarkers in a way it can transform healthcare to patients much quicker by having this complete ecosystem. So when you look at this public policy situation, we think on the left-hand side, the safety of each family, each person, each community versus the economic balance is a real challenge that is trying to be established. Herd immunity is a key trait and a key factor and metric that we're watching to see when do you get to a level of immunity that protects a society. We're nowhere near that in the United States. So there's still a lot of masks, social distancing, cleaning protocols being put in place. But the real challenge is when can we return to work safely, play, and actually return to our lives safely? So testing accuracy and scale are huge challenges for the political world right now. And public demonstrations has further probably created some stress on our overall transmission rates. How do we start to get back on airplanes and travel around? When does entertainment get back to the way it was maybe 6 months ago? When does the sporting community get back? Well, on the right-hand side, science and technology is honing in on the transmission rates, the detection, and can we detect this a lot earlier? Because many of the transmitters have no symptoms. And this is making this particular virus very difficult to manage from a policy standpoint. So the timing of seeing these infections has never been more important to get it earlier. And how can we predict the severity? Some people get it very severe because they inhale it and they get it into their lungs and it gets into the blood very rapidly systemically. And others maybe just get local infection just in the nose and they create antibodies before it gets to the lung. So treatments are key, prevention from vaccines, and the long-term impact on the health impact of fatigued immune systems, as well as a lot of neuronal death, which are these biomarkers can now test for as well, are the long-term impact that we see a lot of providers and payers being very focused on trying to get underneath of and understand. So protective immunity is a key area that's being focused on, and the strength of that immunity and how long will it last. The challenges we have right now is that we're fraught with false positives and negatives. Many people are told they don't have COVID, but they do because there's a false positive, or I'm sorry, a false negative. That they actually have it, but the nasal swab doesn't actually get the infection. And so that could actually further embolden someone to transmit thinking that they're fine when they're not. And so transmission is a key area that needs to be focused on. And the neutralization aspects of what we call the antibody response, or the innate and the adaptive immune response, what is neutralizing the virus is a key area of science right now. Asymptomatic people and populations is a key driver, and even pre-symptomatic detection is something we think these ultra-sensitive biomarkers are going to enable. So how do you quantitate how much antibody you have, how much antigen, which is the virus you have, how sensitive are these tests? Can they be scaled up to a level that can be utilized and have utility cases around the nation and potentially around the world? What we have learned is that there's very few people that have been tested that don't have symptoms. And so getting good sample cohorts of patients that actually have the virus and no symptoms, we think is key to building these validated tests. And we are uniquely qualified right now using this PPH ecosystem to get at these samples, which is key. So I wanted to show this. There's different ways to sample. You got this mucosal local nasal swab and neopharyngeal swab, which we know it's like harpooning almost to the brain, trying to get the infection. It normally requires another operator or a healthcare professional that has to be protected. It's a very challenging test. And you've got saliva starting to come on in some new gumbal CFF that it's like gum fluids that are between the teeth and the gum, which we think could be a big opportunity for seeing this disease earlier and in a way that can be scaled. And then you got the systemic where you look at the blood itself. And many times we think that the virus doesn't even make it to the blood, but when it does, seeing it very early and then seeing it in dry blood spots that can be a simple finger prick at home eliminates a lot of the problems. And you can see down below for the virus testing today, there's a lot of red, meaning that there's a lot of inaccuracies, a lot of high cost and low throughput. We're putting a lot of people at harm's way. And in the antibody testing, there's probably about 24 tests that have been approved. But even the CDC and the FDA are saying, don't make public policy right now based on antibody testing because it isn't understood how much of it's neutralizing, which is another key area of focus. And then ultimately, there might be a possibility of having a combined test that looks at the antigen virus as well as antibody. And that's one of the key areas that we're focusing on as we look at large stage population. So when I make this click, you can see there's a lot of barriers to invasive sampling and false positives and false negatives. And a lot of the solutions that we think are really going to be transformative is having exquisite sensitivity to get to dry blood spots, saliva from home care sampling, and then have the accuracy to digitally understand the actual quantitation and then connect it to the neutralization. We think at the end, this kind of testing build out is going to allow some significant greens, which until you get the greens, I think you're going to have garbage in and garbage out, which could lead to some really difficult scenarios. So when you look at these digital biomarkers, over the last 5 years, there's been huge advances in being able to see proteins in blood at levels 100,000 times more sensitive just where we were just 5-7 years ago. And that's leading to just a profound level of ability to see disease in other categories, not just infectious disease, but to see it earlier, to see heart attacks before they happen, to see cancers before they present symptoms when they're treatable, to see Alzheimer's long before dementia, maybe as much as 15 years when drugs might be much more effective. Doing that non-invasively with saliva and blood dry spots, we think is the future of medicine. So lately they've been being deployed in helping drugs get approved, because if you can recruit patients in with a disease before they have symptoms, it's easier for a drug to be effective at a lower dose, which lowers the toxicity and increases the efficacy. And we're seeing already that if biomarkers are used, there's a 300% increase in the probability. If you get a phase 1 approval, you'll get a phase 3 approval if you use biomarkers as secondary surrogate endpoint, even now several primary endpoints in several neurological categories. So you can see the number of publications are going almost exponential, which is key for really advancing the field credibly, to allow the scientific community to have third-party peer-reviewed publications to validate the technology. That's happening now at a very exponential rate. And the number of digital biomarkers across the body, looking at all these different disease categories, is now up to 330, which is another key area. All these companies now are utilizing these, what we'll call digital biomarkers, ultra-sensitive, to help get drugs approved and for their research. This is a very large community, and this has all happened in the last 4 years. So when we go into COVID, you can see that when you increase the sensitivity, which we just did by the click of this slide, it allows you to see things earlier, see the disease much earlier, see the antigen or see the antibodies, or see the actual cytokine storm, which is what's actually the lethal componentry of the pathology of today's COVID is the cytokine storm. The immune system upregulates to try to protect you, but it gets out of control and it actually drives into pneumonia and is a real toxic part of COVID. These biomarkers are allowing you to see this very much earlier. In population studies, are starting to be deployed using actual home care testing of finger prick, dry blood spots and dry blood sponges to get at some of these key answers, and a development opportunity and the potential we think is endless to not just help with COVID, but then to migrate it right into cancer, into heart disease, and into all these neurological disorders. So the Powering Precision Health Syndicate, as we mentioned before, we have around the world in all the hot zones, major researchers that have been collecting data and collecting samples utilizing these digital biomarker platforms. And that has allowed us to do more and more testing and to actually recruit samples from around the world into the United States to run trials which can be used to take these digital biomarkers into studies that can support an emergency use authorization with the FDA. And we now know that this is an area of extreme opportunity to try to get testing to the level of precision that we know is going to be necessary to get safely the United States back on its feet. So again, when we look at the right-hand side here, this rapid RT-PCR testing, real-time PCR testing, where they're trying to harpoon the infection onto a nasal swab or a nasal sampling technique. There's just a lot of false positives. The DNA and RNA can stay in the blood or in the swab for a long period of time after the actual infection stops shedding, which also then misleads someone that's recovered from COVID that has antibodies to think that they're still got COVID because you get another positive on the actual antigen test. All these things are kind of potentially misleading us into policy that could actually hurt us. So on the left-hand side are some of the areas that we're really honed in on, working hard right now on this dry blood spot. This is a Neoterix device on the left-hand side through some extraction approaches, going into a platform that then can measure these biomarkers. And then we're also looking at the saliva opportunity. This is just some data that says that there's already concordance between serum in these dry blood spots. And this is important for establishing cutoffs and being able to see data. There's already data starting to come out shown here where you can look at the antigen orthogonally, which is the red coming down as the antibodies go up. So these highly precise digital biomarkers are allowing both of these, at least in pilot and in developmental R&D type assays, to reveal this kind of data, which is important for the development. Here's some more cohorts. These actually came from Germany. These were unfortunately, extreme patients that many of them were at very lethal doses of the infection and very delicate with some of them actually presented with cytokine storm. So these levels that you're seeing here from serum and plasma are at higher levels because of the severity of the disease. But we're also seeing NFL, people are losing taste and smell. So neurofilament light is another way to measure in blood the death of neurons. And so we can see as the antibodies come up, concordance with the NFL levels. And so that's another series of publications that are now coming out, is looking for how much long-term impact there is to COVID patients on their neuronal health. So the key now is that there's a lot of focus on neutralization, making sure that the ACE2 receptor on cells doesn't connect up in the top left with this virus. And so blocking that with antibodies is key. And today, anyone that's looking at spike has got, we think, good concordance with antibodies, probably correlating with neutralization. And now there's specific neutralization tests that are being looked at to correlate. And you can see if these antibodies don't stop that, then they're not really helping stop the reoccurrence. So we're actually looking at taking these samples and then quickly moving them into central laboratories that we think are populated around the country, that we could actually put some of these ultra sensitive platforms in it and then get results probably within a 6 to 12 hour period. And we think these are important. And if you look at these labs around the nation, there's these large centralized reference labs that can actually get to almost, in a 75-mile radius, half the population of the United States. So even though it might take a little bit longer to get an answer, you get the advantage of home care, not creating transmission of the infection, and you get to see the answer much earlier, which is key here because of the ultra sensitivity. So we think the digital biomarker brings a lot of advantages that's shown in this slide versus some of the traditional testing. And we're working hard to do that, but then later, longer term, we see trials starting to be run around, does the ACE inhibitor, for instance, actually help prevent COVID. People that are on ACE inhibitors, there's some theories suggesting that they might have much lower incidence. And so getting actual trials using dry blood spots to validate that and get third-party publications to support that is key to advancing the science. And even with NFL, we already know it plays a major role for Alzheimer's, for MS trials, and TBI, Parkinson's. What we want to do now is further understand that with COVID. So in summary, the pandemic is jolting a new reality, garbage in, garbage out with public policy. If we don't get good testing and use this ecosystem, we could end up further exasperating our challenge. So with that, we'll just turn it over back to Laura to see if there's any questions around this discussion.
Thanks, Kevin. We do have our first question already, which I'm really excited about because I actually have done some research on this particular topic. I was looking for a speaker. So I'm going to ask the question real quick, and then I'm very excited to hear your answer to this one. And thank you for that presentation. I learned a lot, and I hope everybody else did too. For better screening measures, what are your thoughts on sewage testing for fecal viral RNA? So there is a company in Somerville that's working on that.
Yeah, so there's different types of testing that the public policy is going to really need to look at. Obviously, the things that you just described do have real potential for what I'll call population level understanding. But in the end, you're going to probably need to get to individual understanding and have some technique that's going to tell a person whether they've got sufficient immunity to protect them from getting the virus a second time. And then how long will that immunity last? I think that that's going to be a very core area of focus, as well as are they still shedding the infection so we can get them out of society during the period when they're shedding the infection. Those kinds of tests won't be coming from the fecal and what I'll call the sewage assessments. Population levels absolutely can, but I think the individual level is going to be key to truly getting the population comfortably and confidently back on their feet again so that they can feel comfortable they're not going to have a recurrence. Vaccines clearly will play a role in that as well, and having good drug treatment so if you do get it, you can feel like you're going to be able to be saved. All of those are factors that we think this testing at an individual level will bring a lot of value, but absolutely, I think the population level is important as well.
Thank you. I did not think about any of the implications of that. Great synthesis, this is the next question, great synthesis of the health and economic impact along with the promise of new tests. So it's exciting. Can you talk more about how you can help advance an effective vaccine?
Yes, you know, the key with vaccines right now is to make sure that they're creating with the vaccine a safe and effective dose of some level of neutralization. And so there's different techniques, different types of vaccines being deployed. And the challenge in these vaccines is getting to larger cohorts because every person is different. And so you want to make sure that you're trying the vaccine across a large enough population to ensure that there's nothing lethal or anything that's going to trigger the actual virus in a way that puts it out of control, or making sure that the vaccine doesn't impair you in any other way. So that the safety of a vaccine is first and foremost. And once you feel like you've got sufficient safety, then making sure that you're creating sufficient neutralization to protect you. And then for how long? Will there be a second vaccine needed? Will you need to boost it? So, did the vaccine take? I think there's a lot of different ways these technologies, ultra-sensitive technologies, Quanterix's Simoa platform is just one of these ultra-sensitive biomarkers that can look at the innate immune system, all the cytokines, to see whether or not they're upregulating appropriately with the vaccine being given. You also can then look at the adaptive immunity, which is the serology, to ensure that the right, IgG, IgM, IgA, whatever that is that the vaccine is trying to create, make sure that those isotypes are effective. And then, I think making sure ultimately that we measure the antigen very early, long before symptoms, so that if the vaccine stops being productive, we in society are going to be able to catch it before there's any kind of transmission levels that start to affect negatively society. So you don't want to have false hope in a vaccine. That's key. And so testing and the broadening of testing, the more testing you can do, the more confidence you'll have that the vaccine will serve you both in a way that's it's safe and effective and plus effective. So that's, I think, the key areas that these ultra-sensitive biomarkers can give you. It's like high definition TV. You'd rather watch a show now or watch sports on high def. And that's what these technologies are doing. They're bringing a level of definition to the answers that allows you to quantitate, that eliminates false positives and negatives. And that's the key, we think, to making sure the safety of not just society for going back to work, but for vaccines and for the drugs that are being deployed to try to help, bring a patient that's got severe case of COVID, back to normalcy. And by the way, the other thing is, how are you going to be impaired longer term if you've had COVID with your neuronal death? There's a lot of immunity fatigue that there's concerns about. Being able to track immunity and watch cytokines from a home care drug dry spot standpoint, we think will be transformative in the future of the way healthcare might be practiced. And COVID has actually allowed a lot of this acceleration of these kinds of techniques that I think are going to play a broader role in the broader medicine post-COVID.
It's like you knew what the next question was going to be. You set it up perfectly, because this is the next question. What lessons have you learned from this experience to advance research for other forms of illnesses and infectious diseases? So post-COVID, what can we use that we've learned during this time?
Yeah, I would say that if you talk to my team at Quanterix or the Powering Precision Health team, all of them are just amazing people that are so smart, so driven. Many of them have not stopped working for 120 days straight. Even Easter Day, many of them worked 12-hour days. So one of the things that we've noticed is that getting good samples and specimens ends up becoming key to getting testing validated. And getting good samples took us months. And we actually had to get them initially from one of our top research collaborators, Ike, over in Germany, who was able to FedEx a bunch of samples that we were able to use to validate and get our testing going. So I think that one learning is, it's really important if we ever find ourselves in this kind of scenario, is really getting testing or specimens so that anyone that's got disruptive innovation can have access to those samples. Obviously, I think we've learned a lot about the testing in our country, the scale of that testing and the precision of that testing. And I think we've got so much mystery with this disease, particularly around what we'll call the asymptomatic, pre-symptomatic, having the ability to transmit the disease, and maybe even given the confidence that they don't have the disease from the testing itself, that bad testing can be worse than no testing. So I think that there's been a lot of focus from the CDC and the FDA to really try to get good testing. And I think one key lesson is bad testing could actually exacerbate an issue. And we're seeing a major relapse in the United States right now. And I would have to say that testing, bad results can actually significantly impair your ability to get back on your feet again. And that's why we're really much focused on making sure we get third-party peer-reviewed validation of every step of the way. Because again, if you mislead the world into thinking you've got something testing-wise that you really don't have, you're really hurting the world, and you're hurting economies, and you're hurting families. And so we really are putting a lot of focus on the precision of these tests.
That's amazing. I know that that's what everybody's worried about, right? All this false positive and so many people feeling like they're safe to go out and live their lives, and they're definitely not. Thank you so much, Kevin. I really appreciate your time this morning. Everyone, we're going to take a break from our Friday Possible Talks for the summer, or at least July. So if you've been tuning in every week, I appreciate it very much. I feel like I've learned a lot over the course of these Possible Talks. I know our CEO, Bob Coughlin's on this morning, and I know he would want me to say that we're so excited about the collaboration of academia, government, and corporations like yours, Kevin, who are bringing everything together with policy moving forward. So thank you for that. And everyone, we won't be here next week, so have a great 4th of July. We'll see you in a few months. Thanks, guys. Have a great day.